Good evening, everybody. I know that not being able to do this in person might be a disappointment for some, but it gives me the opportunity to welcome viewers and indeed our speaker from different parts around the world. My name is Andrew Cuff, and I'm an associate lecturer here at Hull York Medical School, based at the University of York campus. It is my pleasure to be hosting this event tonight as part of the York Festival of Ideas. Before we get started, I've just got some basic housekeeping I have to do. On the technical side, if you're watching live, you can ask questions using the Q&A button on your screen. This is available throughout the event, so feel free to ask questions at any time. Should you have any technical issues, such as the loss of Wi-Fi, you can rejoin the event using the original link. Please also remember that today's event is being recorded, so you'll be able to watch this at any time later. Subtitles are available in the event. To turn these on or off, use the CC Live transcript button at the bottom of your screen. Our speaker tonight is Professor Kenneth Catania. Professor Catania, or more simply just Ken, got his start at the University of Maryland before going on to complete his master's and PhD at the University of California, San Diego. In the years since, he's found himself at various positions at Vanderbilt University in Nashville, Tennessee in the USA. Ken is still at Vanderbilt, where he is now the Stevenson Professor of Biological Sciences. Across his career so far, he's published over 100 papers, several book chapters, and received a multitude of awards, including a MacArthur Fellowship. You are all here tonight to hear about his work, which is covered in his recent book, Great Adaptations. He will talk about a diversity of species from electric eels to star-nosed moles, snakes and shrews, and even zombie-making wasps. Ken has often been drawn to their incredible sensory systems, but sometimes this research takes a detour, which in turn has led to its own extraordinary discoveries. We will show not only how studying these animals can provide deep insights into how life evolved, but also how scientific discovery can be filled with adventure and fun. Without further ado, I hand over to tonight's speaker, Ken Catania.
Well, hey everyone, it's great to be here today, and I want to thank Andrew and the organizers of the festival for giving me a chance to talk about my book and some of the creatures that I've studied in my lab over the last couple decades. And so I want to do a couple things today. And one of those things I want to do is just sort of introduce you to some of the creatures that I've studied over the years, because I think they're just really absolutely fascinating animals. And they present some interesting scientific mysteries that I'm going to talk a little bit about. And then the second thing I wanted to do is just sort of talk a little bit about the way that science is done, at least in my case, how I have sort of approached some of these different scientific questions about these different animals. And then I'm going to dive in to give a couple of more detailed examples about some of these really interesting creatures and what they're capable of doing, and maybe kind of let, in some ways, the animals speak for themselves. So let me start by telling you a little bit about the animals. So one of the most interesting creatures that I've looked at in the course of my studies is called the star-nosed mole. Really kind of amazing animal. It's got this bizarre nose. It's got all these appendages on it. There's nothing else quite like it out there in the world. And so this is sort of a question that's begging to be sort of addressed. You know, what's the star for? And how is it used by the animal, and what abilities does it give the animal? So I'll tell you that story in some detail and how I got involved. And moving along to sort of another mammal, this is a water shrew. And this is not a rodent, even though it looks a lot like a rodent. It's a shrew, which is sort of part of the historical group of what are called insectivores. And it's a really aggressive predator, as you might guess from this image, as it confronts this crayfish and is about to overcome the crayfish, and that'll be the shrew's dinner. So another really interesting creature. Moving to some reptiles. So this is a tentacled snake, and this animal is a unique in the same way that a star-nosed mole is in terms of having these sensory appendages, but it also has some behaviors that are some of the sneakiest things that I have ever seen any predator on Earth do in terms of how to capture prey and essentially scare prey to death, in a sense. And so that's another one of the stories that I sort of address. The electric eel is a fascinating animal that has been studied for centuries, and yet we are still learning to this day new things about this amazing animal. And not only how it produces electricity, but how it uses electricity in some incredible ways in terms of how it can interface with other animals. And I'll talk about that in some detail. The emerald jewel wasp is an insect, and it has a famous ability to essentially create zombies out of cockroaches as a way to find a host for its young. And that story is tremendous in terms of the ability of this particular creature to manipulate the nervous system using targeted venom placed in different places in the cockroach nervous system that essentially bends the cockroach to the wasp's will. And that's another really fun story. And actually this one, I'm not going to even tell you what this one is. I'll leave that if you want to know what's going on here with this alien looking thing. That's a real bit of biology. For that, you'll have to look into the book. So that's a little bit about the creatures, just a sampling of some of the ones that I talk about. And now I want to talk a little bit about what I sort of call doing science. And I want to give a metaphor or analogy of the way it has worked for me. And for that, I want to refer to the Hubble Space Telescope. And that might seem like a strange place for a biologist to sort of look for an analogy. But I like to use this example because we think about the Hubble as a way of taking a look at what's going on in the universe, and often because of that, discovering amazing things, even when the Hubble Space Telescope has been aimed in an area that we might not think is interesting. And so I like to use as an example of that, this ultra deep field image from the Hubble telescope that revealed in an area, a little sliver of supposedly blank part of our universe. It revealed 10,000 galaxies and completely revolutionized our view of the world. And I think that happens in science, biology often, where you may look at something you don't think is necessarily interesting, or you don't know why it might be interesting until you take a close look. And then you'll discover things you never knew were there. And having used this analogy, I now want to sort of literally bring things down to earth and extend the analogy a little bit. So this is a pond that I visited when I was teaching online for the students last semester. And what happened, and what often happens to me, sort of expanding the analogy a little bit, is I saw something move in the middle of this screen, or image, I should say, and I aimed my binoculars there. So I'm going to show you a real image of a closer up view. And so this is actually, I used a telephoto lens to take these different images, of course. And actually what happened when I looked through my binoculars was I noticed this turtle that I never would have seen had I not viewed this scene through the binoculars. So again, sort of like looking through a telescope, but this being a little closer to home in terms of biology. When something draws your attention, you never know what else you might find in the image. And that truthfully happens to me almost every time that I study a new species. And that's some of the things that I want to talk to you about today. So sort of moving into the actual things that I've looked at, I'm going to start with a part of the book that I have labeled the snipe hunt. And the snipe hunt, I'm referring in this case, this is talking about the research into star-nosed moles, which I think are a good stand-in for a mysterious animal that you may have a lot of trouble finding. And so let me go back to telling you a little bit about star-nosed moles. So this is actually a rodent-sized, sort of a little bigger than a mouse-sized mammal with this extraordinary nose, which is not used for smell. And I'll tell you what it's used for as I move along. But I want to tell you a little bit about the sort of personal story of how I got involved in this. And that involved me having a chance to volunteer at the National Zoo in Washington, D.C. when I was an undergraduate student. And that volunteer experience was revolving entirely around the star-nosed mole, because they were studying the star-nosed mole at the zoo and they needed to get more animals for their studies. And they basically, I volunteered to be involved in this research. And then eventually I was the guy that had to go try to find more of these animals. And this was sort of an epic quest to find this rare creature, not endangered, but very hard to find. And I went up to an area in Pennsylvania and searched around in the habitat. And I should say star-nosed moles live only in wetlands and sort of muddy areas in the wild that are where it's really hard to find tunnels. And part of the fun story of that experience for me was eventually, after driving around for hundreds of miles, I found a wetland that looked perfect for where this animal might live. But I was really worried because adjacent to that land was this cabin, and you might sort of use your imagination for stumbling onto a cabin on someone's land in a sort of remote area, and thinking, oh, well, should I go on this person's land? Should I even approach the cabin? I was worried about who might live there, they might think about me being on their land. But it turned out I did eventually get up my courage and go and introduce myself to the owner of the land. And it was this guy, Carmen, who was a wonderful, supportive fellow who, when I told him that I was on this quest to find these animals, he said, "You can use my land." We got to talking about the biology. He was an outdoorsman who did a lot of fishing, as you can tell from the image. And we became friends over the years. And I always went back to this same area, and he would let me use his cabin and allow other people to come up there and sort of play poker in the evenings and hunt moles and set traps during the day. So that was a really nice sort of warm reminder that you shouldn't let your imagination get carried away with what you think people might be like. And I think that applies a lot to what's going on in present day things. So our imaginations about what people are like are a little dangerous, and it's always good to get some personal interactions when you can. All right, so that's how I ended up working on a remarkable piece of land, and I'll tell you how this sort of collecting works. So you set hundreds of traps into the wetland area, and this is an example of these little live traps that you set in the tunnels where these animals live. And I didn't really know at the time what I would get when I started doing this research, but it was this amazing example of learning about the diversity of small mammals that live in these little tunnels. And I just want to show you some pictures of these animals. So this is a rodent. This is a little vole. There's huge numbers of voles in the area. Sometimes I wonder why the world isn't made up of voles, given how many I caught. This is a shrew, very distantly related to the rodents, and it actually has venomous saliva. One of the few mammals in the world with venomous saliva. This is a short-tailed shrew. This is a pygmy shrew, and these are among these, I'm sorry, this is a masked shrew. This is a masked shrew. These are among the smallest mammals on the planet, only weighs about 3 grams. So that's about the weight of a penny. Incredibly fast, really interesting nervous systems. And I talk about what their brains are like in the book if you want to know more about that and what gives them some of their amazing abilities. This is a hairy-tailed mole, which you can guess has a hairy tail, and it lives in the same areas as the star-nosed mole. This is the water shrew that I was talking about, beautiful animal that can catch fish. And this is a short-tailed weasel, one of the main predators in the area that basically eats all these other small mammals. And finally, I was able to catch star-nosed moles. And that was a very fun experience to eventually realize that out of all these mammals, in different traps in different places, I was able to actually find this legendary animal and bring these guys back to the zoo, where we started to conduct a series of studies. Now, I'm going to leave out some of the ups and downs of that research and sort of tell you the eventual success story that ended up happening, which was to very much, I should say, sort of use the example of taking a closer look. So one of the things that happened early on was sort of theorizing about what the star might be for. Didn't work very well. What did work very well was to take this metaphorical approach of simply starting to look more closely instead of getting very sophisticated with experiments. The first thing was to do descriptive stuff. And now I want to tell you about some of those descriptions, because they are some of the more important things that you can do in science. And this is where things got really interesting. So here is the star under the scanning electron microscope. And this is an amazing piece of anatomy. in terms of what this star is like. It's only about 1 centimeter across, so it's about the size of your fingertip. But as you might notice, and let's see if this cursor will work for you. Hopefully you can see this arrow here, that the star is made up of these appendages that are completely covered with these little domes. And these domes are sensory organs. And I'm going to zoom in on this for you. And these are the nostrils here on either side. And so I want to just sort of reemphasize this special structure, the star, is not about smell. It's about these special sensory organs that are called Eimer's organs. And you could actually think of the star as entirely made of these sensory organs called Eimer's organs. And so, of course, the next question is, what are Eimer's organs for? You might wonder about that. That's sort of a key to what is the star for. Well, it turns out our studies showed that the Eimer's organs are incredibly sensitive touch organs. They're little tiny touch domes that give an amazing acuity and high resolution to the star. And I'll tell you a little bit more about that and show you some of their behavior. But I did want to also just sort of emphasize that the sort of comparison between a human hand, which is a very sensitive touch organ, and the star-nosed mole. So a human hand has about 17,000 touch receptors. So that's how many you would typically have in your fingertips and palm. And now consider how a star-nosed mole's nose compares to that. There's actually 100,000 touch nerve fibers compacted into this little structure. And this is actual size compared to a human hand. So there's more than 5 times the number of touch receptors concentrated into the size of your fingertip in this animal. And then you might wonder, well, okay, what can a star-nosed mole do with this 5 times more touch receptors? And for that, I'm going to show you a movie. And I'm going to sort of jump out of the screen here and just let you see that full screen so you can see it in better resolution. So, but first I'll tell you what you're going to see. You're going to see an aluminum foil piece. And then I've videotaped a star-nosed mole coming out onto the aluminum foil. And the mole has been trained to detect a little tiny X mark on the aluminum foil. And when it detects that X mark, it's going to break through the aluminum foil and get a food reward. And a little laser will come and laser pointer will show you where that X is marked. So let me play that video for you. Okay, so there's the laser pointer showing where the little X is, and here comes the mole to find that. Remember, it's been trained to do this. And look how quick it's moving the star, touching really rapidly. And there, it detects it with about 1 touch and breaks through the aluminum foil. There's a little piece of an earthworm down there that it's really happy to get. So that was a good example of something the mole can do without using the sense of smell, because the food was under the aluminum foil, just using the sense of touch. And I'm going to show you a little more about that in a minute. Okay, so now you've seen a star-nosed mole in action, and you've seen the anatomy of the star with those little sensory organs that cover all of the appendages. And now I want to tell you a little bit more of the story of the way science often unfolds, at least for me. And that is, there's often little seemingly inconsequential cues that may actually turn out to be really important in sort of solving a mystery, or at least leading to another mystery. And I want to tell you about one of those. And this is related back to the anatomy of the star, something that you might not think of as very important on the surface, but is actually really important. And now I'm talking about kind of descriptive studies that seem kind of simple on the surface, but may reveal something really important. So, okay, so what am I talking about? So here again is the star under the scanning electron microscope. Here is the appendages. There's 11 on each side. And here I'm pointing out these little subtle borders where there's no sensory organs between each of these appendages. And that might seem inconsequential. And yet I'm going to point out a quote by Pasteur about chance favoring the prepared mind. And for me, this little border reminded me about something interesting about brains. And I want to tell you about that next. And I'm going to start with the human brain. And so this is a brain that, in a sense, we're all familiar with. And this is showing you the neocortex, the outer folded sheet of tissue that is a characteristic feature of all mammals and is really enlarged in, of course, us. And one of the features of these brains is that they contain maps of various senses. And so I'm going to talk about the map of touch and just point out where this is in humans and a little overview of what it's like. So half of the body projects to each half of the brain. So of course, I'm showing you only half of the brain here. And it turns out to be the opposite side of the body that projects to, for example, this is a left hemisphere, so it's the right side of the body. It's just a quirk of the way these maps work. And this map is always oriented such that foot, leg, arm, hand, and face are mapped into the brain in this order, meaning that this is where the cells that process touch for the foot are. And for example, this is where the cells that process touch for the face are. Now, one of the interesting things about mammal brains is that all mammal brains have this map of touch. And this is well known in mice and rats, which are commonly studied in neurosciences. And there's a really interesting feature of rodent brains, and I'm going to tell you a little bit about that. So here in this upper picture is their so-called somatosensory or touch cortex with the map of their body. And the large part in the case of the rats and mice is the whisker representation, because that's really important to them. And it was discovered in the 1970s that there is a really interesting anatomy in this area of the rodent brain. And I'm showing this to you here. These are little circles in the neocortex that are called barrels. And each one of these circles corresponds to a single whisker on the face of the rodent. And it's essentially as if, in the case of humans, for example, it would be like you could see a hand in your brain in the hand map, which we can't see for humans, but we can see an anatomically visible whisker map in the rodent. And there's whole books written on this and thousands of papers, literally. These are called cortical barrels. And so when I saw the subdivisions on the star, I wondered, could there be a similar visible brain map in a star-nosed mole? And I probably wouldn't be telling you this story if there wasn't, so I'm going to go ahead and show you that, because it turned out there was. And so this is a really interesting bit of brain anatomy in a star-nosed mole. So it turns out this dark area here in the mole's brain is in fact the map of the star. It's only half of the star because the other half is on the other side of the brain. And this is sort of a newly discovered visible brain map. And this, again, led to other interesting findings. And sort of it's like a breadcrumb trail of discoveries that often occur. So let me tell you what happened next. And that was, well, let me first show you this map a little closer up. Here's 11 subdivisions in the brain, and they correspond to the 11 appendages on the star. And the interesting part, as soon as you see this map, there's an interesting part of it. And all of us can see this. You don't have to be a neuroscientist to notice that this part of the map is huge compared to these other parts of the map. And yet it's not that big on the star. So that raised another question. Why would this subdivision be so very large in the brain, even though it's not very big on the star? And that's a question about behavior, or at least that's one place that would lead you, is wondering how does the mole maybe use this appendage differently than the others. And that turned out to be really interesting. And for that, I want to tell you about what we humans do with our eyes first, because we have a central area of our eye that we use and move around to examine things in detail. And so we're all doing this. You're doing this right now as I move this eye back and forth. You're shifting your eyes. So there's a little bit of sort of things get meta there in terms of this analogy. And this is sort of like moving a sensory flashlight around to examine things in detail. And it's a way to efficiently organize that part of the brain and the sensory surface of our eyes. And a star-nosed mole does the same thing. And so if you didn't move, I'm going to show you an example. If you didn't move your eyes around, for example, let's say you looked at this letter G here. Without shifting your eyes, what you're really seeing is a very blurry, low resolution surround. And it's only by shifting your eyes that you can see this whole book cover in detail. And that's the same for a star-nosed mole, as it turns out. It examines things in detail with that 11th appendage. And now I want to show you that in a movie so you can see these movements. This movie is going to be very much slowed down, slow motion, because the mole moves super fast. And so let me just move to that movie so you can get an idea of the mole's sort of so-called foveation movement. Okay, so this is filmed from below on a glass plate. Here's the first touch to that little bit of food it's interested in. It actually does a double take, moves away, and then comes back and touches with the center part of the star to make its final decision. Okay, so what you just saw was the mole often will touch things with these outer appendages, but it always moves to this 11th appendage before making a decision about whether something's worth eating or if it's interested in something, exploring it in detail. And of course, you do the same thing with your eyes. You see something interesting in the peripheral vision, you immediately shift your eyes. So it's really a pretty remarkable analogy for the way the sense of touch works in a star-nosed mole and the way that our own visual systems work. And that's somewhat surprising considering moles are essentially blind, that you would find such a sort of similar organizing principle for high resolution through the sense of touch and the sense of vision. Now, I mentioned sort of things fall out of these studies, and another thing did end up falling out, so to speak, a clue to something interesting going on with star-nosed moles. And that simple clue is that I showed you the last movie in super slow motion so that you could see the movements of the star. And that might make you wonder, and it certainly eventually made me wonder, well, what's going on with speed and the star-nosed mole? Why is it so fast, and just how fast is it? So using these very high-speed cameras, we were able to record how quickly they can use the star, foveate, detect things, and eat. And now I'm going to show you a star-nosed mole in real time. And this is going to be a video that loops, again, a mole feeding on a glass plate, just so that you can see how very quick they are. It's pretty incredible thing to see. So let me show you that one. Okay, again, this is filmed from below, and it's going to loop so you can see it multiple times. How quickly this animal can eat these 8 different tiny little things. It's truly amazing. Okay, I hope that gives you an idea of just how very fast this star-nosed mole is. In fact, I have trouble not using words like incredible and amazing and remarkable too many times when I talk about this animal, because they actually are hard to believe in terms of just all the things that they can do. And it turns out when we published the results of that high-speed eating, that actually put the star-nosed mole into the Guinness Book of World Records as the fastest foraging mammal. And if you can believe it, what I've told you so far only scratches the surface, no pun intended, about what these animals can do. There is a lot more literally to the story. So if you want to learn more about them, I talk a lot more about other things that they're capable of doing that sort of fell out of just observing their abilities and what they're capable of. And now I'm going to sort of turn the page, and I have this video to remind me that I'm metaphorically turning the page to a new chapter to tell you about another animal that I think is one of my favorite creatures on the planet. It's the electric eel, and these animals can do some really interesting things. So here's an electric eel. This is one of the eels from the laboratory. So I started out doing photography with them because I just think they're such interesting creatures, and I wanted to show some imagery to my class. And I ended up discovering, as usually happens, some really new things about what this species can do. Now, just to orient you about this animal, so the reason that this animal lives in South America, and it's a fish, even though it has this long sort of eel-like appearance, it basically has most of its body containing an electric organ, and that's why it can be so powerful. A large one can give off up to 600 volts. And I should mention that it's a freshwater species. They live in the Amazon region. They live in South America. They evolved from weakly electric fish, and they ended up evolving this massive electric organ that they can use as a weapon. Now, most weakly electric fish use electricity only to sense their environment and not to defend themselves or to hunt with in terms of an attack strategy, whereas eels obviously can use it for both offense and defense. And they give off their electricity in pulses in the water. And when they're giving off these pulses, essentially a dipole field surrounds the animal. So this is what you're seeing here with these lines, sort of the where current flows around an electric eel as it's giving off its electricity. And the electric eel, you can imagine, was known about for centuries because it's giving off such high voltages. And it has a lot of interesting historical links to science and discovery. And I'll just mention a couple of those for you. And one is that Volta was actually inspired to create a battery based on the anatomy of the electric eel. So this invention that we all depend on every day, and I am using right now, and many of you are, was inspired by the anatomy of an electric eel. And Volta actually called it his battery an artificial electric organ, similar to the natural electric organ of the torpedo and the trembling eel. And the trembling eel is the electric eel. That was in 1800. Another amazing thing that happened in 1800 comes from another famous historical figure in science, and that's Alexander von Humboldt. And he went to South America to collect electric eels to try to better understand what electricity was. And he tells this fantastical story about how the fishermen collected the eels for him when he hired them to get the eels. And it basically involved herding horses into a pool containing electric eels. And according to the story that wasn't believed by everybody, the eels attacked the horses, shocked them repeatedly. And here's one of very many illustrations of that event as the eels, you can see one down here swimming around the horse, shocked the horses. And the strategy was that the eels were exhausted by this battle with the horses, and then the fishermen could catch the eels that were no longer giving off electricity and provide them to Humboldt, who did publish experiments that he did on the eels in South America. So I'm going to come back to that story because there's some really interesting links that I want to tell you about that I think will be entertaining. But now I want to tell you a little bit about electric eels and some details. And one of the things I want to tell you is that the electric eels have two power settings. It's almost like you can think of them as having high beams and low beams. Their low beams are their weak electricity that you would not sense if you were around an electric eel in water. And then they also have this high voltage, which is the weaponized output, the very, very powerful electrical pulses that they give off when they're swimming around hunting. Okay, and so now what I'm going to show you next is a video of an electric eel basically swimming around. I'm going to enlarge this video, and you're going to hear some noises. And what I want to make sure you're aware of is those noises are coming from a speaker that has wires attached to the water. So electric eels don't make noise, but first you'll hear an electric eel giving off these pop, pop, pop sounds, and that's the sensory side of its electrical output that's low voltage. And then you'll see a fish up here, and you'll see it attack and eat the fish, giving off the high voltage. So let me go ahead and play that for you. All right, so that was an eel in real time, basically sort of giving off its normal different high voltage and low voltage pulses. And where things got really interesting was to put the electric eel under the slow motion camera as it preyed on fish and see the interaction between the eel and the prey. And so what I've done for you is to colorize the frames red during each high voltage pulse. So you can see the relationship between the eel's output and what happens to the prey fish. So let me go ahead and play that for you. Okay, don't forget this is slow motion and the fish is actually moving very quickly, but then it's completely frozen up once the high voltage starts. Here's another example. This is a fast moving fish, and once the eel's high voltage starts, it's frozen like a statue. And here on this last one, notice that when the eel stops giving off the pulses, this immobile fish that's completely frozen starts to swim again. So what's happening is temporary. Okay, so that was an electric eel inactivating prey very quickly. And the question is, what's going on here? All voluntary motion was basically arrested in those fish within 3 milliseconds of the onset of the high voltage. And how could it possibly do that, especially since it's temporary? So that last fish you saw started to move again as soon as the eel slowed down. And so the idea of what might this be, how could this work, was essentially analogous to a taser, a law enforcement taser, which basically causes muscle contractions by sending pulsed electricity. So pulsed electricity is what an eel sends out. The taser uses these wires, of course, from the darts that get sent out into a person, and that makes it impossible for a person to move. So it looks like essentially, and I'll just say that the study showed that yes, in fact, an electric eel is acting like a taser. So electric eels invented the taser long before we did. And they have an advantage that they don't need wires. So essentially the water around an electric eel is acting as an antenna so that all of the animals that are within range are being affected by the pulsed electricity and having their muscles activated. And it truly is a sense of remote control by the eel. So the eel is able to control the muscles of nearby animals in a fairly sophisticated way. So, it can just freeze them up, as you just saw in that slow motion, but it has other tricks as well. So if an animal is hiding, the electric eel can give off little blips of high voltage, and that'll make the animal twitch, and that gives away the position of the animal to the electric eel. So it's a really amazing kind of insidious trick. So they use electricity in lots of interesting ways. And now I want to tell you about one other way that was really very surprising that they are able to use electricity for. And this is to actually leap out of the water for an electrical attack. And I discovered this completely by accident by trying to move eels back and forth in aquariums with a metal net. And it turns out, well, if you're wearing a glove, you're not going to be affected by the high voltage at all. But the eels seem to interpret that net as a living thing, and they come out of the water to attack. And that actually was really convenient for sort of exploring why might they do this. And you can set up an experiment. You're going to see that this video shows you an eel coming out of the water, and this is slow motion, and the red pulses again show you the electrical pulse timing as it attacks this piece of metal. And what I was able to do is basically very easily show what you might expect, which is as the eel comes out of the water, more and more power is directed towards whatever the eel is targeting with that leaping attack. So that's a really very interesting sort of behavior. And I wanted to explore this further. And this is a fun part of the story, really, because I couldn't figure out how would I best illustrate this behavior for people. I mean, I can show you this plate here, but that's not that much fun. It's not very intuitive for what this might mean for an animal being attacked. And I eventually decided to use a prop arm. And I thought what's the most economical way to do this? And I eventually settled on zombie arms, which caused a lot of confusion when people looked into my lab and saw what looked like these severed arms piled up in, on the other side of the door. But what I was doing was trying to design various ways to illustrate how an eel would activate the nerve fibers, and in this case, the pain fibers in a nearby animal as it came out of the water, and do this efficiently. And so what I was able to do is put diodes into these fake arms, and I wanted a lot to practice on because it never works right the first time. And so I'm going to show you a movie of the result of that, which I think is very good for getting across the power, so to speak, of an electric eel and defending itself if you were to end up in the water with one of these electric eels. And I should say at this point that this is a defensive response. So eels swallow their food whole. They don't take bites out of things. They don't attack horses and eat horses. They only can eat things they can fit in the mouth. So whatever you're seeing in terms of this behavior is not hunting, it's defending itself from a threat. So let me show you how good they are at doing that. So that's a pretty daunting behavior, and I thought that armful of diodes really brings home just how efficient the eel is at deterring potential predators. But better than that, people didn't believe necessarily that Humboldt's story was true. There were a number of people who said this could not have happened. He must have made it up, because there was no evidence of eels attacking large animals like that. But that's exactly what they do. And in fact, now it's been over 200 years since that, and I was able to publish a paper that basically supported Humboldt's account of what happened. So this is what I showed you, an eel coming out of the water on a plate. It turns out that since I did this study, there was someone who got a cell phone video of a person being attacked by an electric eel in a very similar manner, just like what was going on in the laboratory. And I was able to find an illustration by a friend of Humboldt's in a fairly reclusive journal that shows the exact same behavior happening with the horses. So this is published in the 1800s by someone who knew Humboldt. So it really looks pretty clearly like what Humboldt talked about was totally true. And I was just so nice to be able to support sort of an account of this really strange behavior from a famous scientist from over 200 years ago. That was one of the most fun studies I was able to do. And the last thing I'm going to tell you about it relates also to this behavior. It turns out, you can start to calculate just how much power can an eel communicate. It's an interesting circuit, and I'm going to show you some of the circuitry related to this. What would happen if an eel came out of the water? Well, it turns out you can calculate all of these different variables using various physics techniques for finding out the resistances of water and of the eel and of the internal battery for the eel. The problem is you don't know how much current flows in this circuit unless you know this resistor. So after spending a long time working on this, I decided that I would sort of take one for the team and find out if these calculations were accurate by using my arm for this. Now, I should point out I used a very small electric eel, so it wasn't dangerous, but I did find a way to measure the current that would flow from my arm through this water container. And this is actually a result of that experiment. And here's the video camera and all the details. But I'm sure you're more interested in maybe seeing the video of this than in actually me outlining how each of these different aspects of the physical experiment work. So why don't I just go ahead and roll the tape on me sticking my arm in and having this small eel come out so I could calculate the full circuit. So I'm going to go ahead and play that. All right, well, with that shocking ending, I want to thank you for listening. It's been great to have a chance to tell you some of the stories of the research I've done. I also want to thank Princeton University Press and especially my editor, Allison Collette, for the wonderful production of the book, and Sarah Henning Stout for helping to get the word out. And I also want to thank the National Science Foundation for supporting my research, and everyone at the York Festival of Ideas for putting this wonderful venue together. So if you have any questions, I'd just love to talk about some of these creatures. All right.
All right. Thank you very much, Ken. I'm sure everyone else found that a fascinating talk. I think there's a few questions already available. If anyone else has some more, feel free to keep putting them into the Q&A box down the bottom. So the first question comes from Rosie. She wants a hint at what that weird creature was right at the very beginning and the story behind it, if you're willing to give any little details out about it.
Sure. I won't keep that as a cliffhanger. So that is a star-nosed mole embryo. So one of the clues is that nose, but they are some of the most unusually developing animals, like everything else about them, completely off scale in terms of bizarre. The star develops unlike any other set of appendages, and appendage development is something that's quite heavily studied by developmental biologists. And yet, just like everything else the star-nosed moles do, they have a different way of going about it. And I talk about that in the book, and there's a whole story about field trips there that as I went on a quest to find what you might call the missing link for the evolution of where the star came from. And I did, in fact, manage to find a missing link type. By that, I mean an animal, a mole, that had an intermediate type protostar. So there's lots of interesting tendrils of biology that, no pun intended, that come out of the star, so to speak.
I guess following on from that, it's a very good question linked to it. Who are the star-nosed moles' closest relatives, and what do their noses look like? Do they have similar brain maps?
Yeah, so that's a very interesting question, and we have pursued that. So the other relatives are the other moles. There's actually about 30 species of moles. The European mole is pretty common, I'm sure, and it turns out that the other species of moles have 2 maps of their nose, and the star-nosed mole has 3. And it's actually a discovery in this species that supports an idea in the neurosciences that one of the ways that you get behavioral complexity is not just to get a bigger brain, but to add more maps. And human brains are full of brain maps, hundreds, probably over 200 brain maps of different kinds, of touch, of vision, of our auditory systems. And yet it's hard to find a specific case where a brain map has been added and you can kind of trace how that probably happened. But the star-nosed mole is one of those cases, because the big difference between it and other moles is its star, and the big difference in its brain, or one of the big differences, is having an extra map of touch. And so it does provide one of these general clues to what's going on.
So moving from moles onto those eels, how does an electric eel avoid electrocuting itself if it's conducting through water?
That is a great question, and one of the big ones that people still wonder about to this day, and one that I wonder about. The one thing that is known is that its nerve fibers are really heavily insulated. So we all have an insulation called myelin that surrounds our nerve fibers, and that actually sort of is a fatty insulating membrane that helps with nerve conduction. But in the electric eel, people observed decades ago that their myelin was super thick. And so that is one of the things that's protecting them. And I think the other thing that's probably protecting them, that's sort of an open question that we're hoping to solve, is the opposite type of biological substance that we haven't yet found, which is probably a path of low conductance that takes the electricity from the electric organ to the water inside the eel's body. And so I bet that that's there. But there's still a lot to do to discover that. And who knows what finding such a biological substance might mean for, how it might be used in other ways if it exists.
I guess there's a fun follow-on from Walter, I'm assuming is a fairly enthused young child. Were the zombie arms real human arms, or are they all plastic?
They were all plastic, easy to obtain, but and I talk about this in the book. At one point, because they were painted to be like zombies, in order for me to make them for the experiment, I had to scrub off paint. And I was in there with a pile of arms in my protective chemical hood in the lab when a student came up to get a form signed, and she looked in the hood at this stack of arms. And of course, I'm kind of working in a medical center type building, and let's just say it was an awkward moment for everyone.
Whilst we're still on the eels, we've had another one come in. Are you able to kind of build upon the process of evolution that resulted in eels having the ability to electrocute aggressors?
So you mean maybe making a better taser? I'm not sure exactly.
I'm guessing the thought process here is, how do they go from being slightly sensory electric fish to being able to use it in not just a hunting mode, but also a defensive mode?
That is a great question that I wonder about a lot myself, and isn't really known, except here would be some thoughts. I didn't manage to have time to cover this, but it turns out that the eel uses the high voltage also as a sensory system for probing its environment. And this is something other weakly electric fish do. And I mention that because you can increase the range of your sensory system by getting higher and higher voltage. So that's one way you might have an adaptive way to increase the power. And so it would be useful to have further and further range. And if you continue to increase your sensory range, you might end up evolving something that, as a consequence, was shocking other animals. And the other way that it might evolve is actually electric fish, even the small ones, use electricity to communicate, and some of them establish dominance based on their electric organ output. So you might have a competition of kinds between especially the courting males that would evolve higher and higher powers, and that might eventually become something that could be used as a weapon as well. But really, we don't know exactly how it happened.
So a final question, I'm hoping, on eels. How painful was the electric shock? Everyone wants to know, I'm sure.
Yeah, so I would say anyone who's worked around a farm and ended up accidentally or maybe on purpose touching the electric fence, it's a lot like that. It's pretty painful, and in fact, it might look wimpy, like I'm pulling my arm away because I'm a wimp, but actually, the eel will activate your involuntary arm withdrawal reflex. So there's no way for me to keep my arm in there, because it sort of switched to the neuroscience side of that. My spinal cord is telling my arm to get out of there before I can even cognitively do anything about it. So it's a lot like touching a hot stove. Suddenly your hand is back here, and then a second later, you're thinking, oh, that hurt. But your arm is back before you actually process that consciously, and an electric eel will do that to you. So that's a good benchmark of pain, but it doesn't do any damage. So, I wasn't doing anything crazy there, and I wouldn't do it with a really big electric eel. It was a small electric eel.
I was going to say, there was the video that you talk about a little bit of someone being zapped by a big one, and that did look a lot worse.
It was a lot worse for the guy who fell over. You can find that on YouTube if you search for person shocked by electric eel. I will say that person got back up again, and I wouldn't show that whole video because they did end up killing the eel. That's what they were trying to do. But the eel at least got a good shot in on the way.
So what first inspired you to get into this area of research, and have you always loved animals?
Yes, that's pretty much exactly what it was. And I do talk about those early experiences in the book. I was someone who sort of wandered around the lakes and streams growing up, always excited about snakes and turtles, and before that, insects and cicadas, and basically any creature that I could find, I found fascinating from a really early age. And I always think that's sort of a good lesson about one of the reasons that we want to preserve wild places, because that's how biologists often get their start. It certainly was for me.
So what's the furthest you've traveled, or the most extreme environment you've been to in search of some of your animals?
Well, I have been to South America and Australia, but not for the research. When I'm there, I'm always looking at the creatures. The crocodilians in Australia are really something compelling, and we have looked at crocodilians in my lab, but I haven't gone on those kinds of adventurous field trips to other countries with the purpose of doing research so much as just to enjoy what's around in Africa and Australia and so forth. And those are just wonderful experiences. But in terms of field work, mostly it's been in the continental, all of it has been in the continental United States, in some of the swamps in the south, and some of the wetlands in the north, in the deserts in the west. So that's sort of covers that area.
I say, may not be their expectations for extreme, but always very interesting no matter where you go. There's always some cool and wonderful animals, aren't there?
Yes.
People are still on these electric eels. Obviously, you've sold them on your love of them. So if a person who has a pacemaker is shocked by an electric eel, does it give them a bigger shock? Does it reflect back on the eel? I suspect I could take a guess at that, but I would love to hear your thoughts on it.
Yeah, I don't know. And I would think that if you had a pacemaker, you would want to avoid electric eels pretty carefully. Now, I'm always cautious about how I talk about this. I don't know of a single documented case that's actually been shown to be true of a person being killed by an electric eel in hundreds of years. So in that sense, they're unlikely to, and they didn't kill the horses outright in Humboldt's case, but they did shock enough that some were exhausted and apparently drowned. And I do think that's the most dangerous thing that could happen to people swimming near electric eels. If you were frozen up, you could easily drown from that. But their shocks don't seem to penetrate the chest to the point of being able to affect the heart directly. But I would still avoid getting shocked by a big one, because you wouldn't want to be the first documented case, right?
I think we've got time for maybe one or two more very quickly. So what do you do with animals once you've caught them?
Well, usually we house them in the lab, And basically, a lot of the times at the end of the studies, we can farm them out. So for example, the electric fish, or I've studied tentacled snakes and just ended up at the end with a lot of pet tentacled snakes at the end of the studies. Or if it's fish, for example, those can often be farmed out to students who are excited about tropical animals.
It's always good to hear. Are there any animals that have evolved a defense, either by detecting low voltage scanning or protective coating of some description, that would prevent them being shocked by these electric eels?
I love that question because we don't know the answer to it, but I think that it's likely that there are. And that may, in fact, be one of my future questions in the very near future, is to try to look and see, are there animals that have managed to defend themselves electrically from electric eels? Right now, we don't know the answer to that, but there's a lot of people interested, and that goes all the way back to people need to go to the Amazon and find out what's going on there. So maybe that'll have to be on my list of field trips.
I'll do a quick shameless pitch for you, but I've got your book. There's something that people might get curious about if they can see. So this is on the electric eels. There's this little QR code. Do you want to just explain that one to people, please?
Yeah, thanks for pointing that out. So I wanted people to be able to see the animals in action. And so that little code, all you need to do is aim your phone camera at that code, and it'll automatically take you to a movie of the animals in action very easily. And I wanted to do that because, like I said over and over again, I think they're amazing, and there's nothing quite like seeing the animal right after you hear a description. Oh, it can do X. Well, you get to see X when you scan that code. And so that was an experiment for the book, and I hope that is fun for people to see and watch themselves.
We can do one very quickly. You mentioned shrews with venomous saliva.
Yeah.
How dangerous is this venomous saliva, and why is it rare in mammals?
So it's not known to do much more than make the local area very painful for about a day. There's actually not a lot of reports of people being bitten by the short-tailed shrew, and I've avoided it. It does seem like the main purpose of that is to paralyze insects. So you can imagine, compared to a snake that has evolved a much more powerful venom, these venoms for shrews that paralyze crickets and katydids and earthworms is much weaker. The only other animal I can think of that's venomous is the duck-billed platypus males have spurs that are apparently incredibly painful, that they use to fight with one another with over mates.
All right. I think, unfortunately, we're out of time for any more questions, but I'm sure on behalf of everyone who has watched, I'd like to thank you, Professor Ken, for an incredible talk. And I really enjoyed the Q&A sessions after. Thank you, everyone who sent in questions as well. They were all very, very interesting ones to have. So the recording of this event will be available on the festival YouTube channel after the show, after the 20th of June. It can be accessed by the Watch Again feature on the festival website, and you'll be contacted by email when the video is available to view. If you'd like to purchase your own copy of Ken's book, the Great Adaptations are available from our partner bookseller, Fox Lane Books. For more information on book sales, please see the festival website or head direct to foxlanebooks.co.uk/festival-of-ideas. And we hope that you've enjoyed the Festival of Ideas. Thank you for engaging with us. Please check out the website for full details of all the events in the festival program. We'd love to hear your thoughts on these events and continue the conversation on Twitter using the hashtag #YorkIdeas. And I'd like to thank everyone again for attending, and thanks once more, Ken, for presenting. It's been brilliant.
Thanks, Andrew. This has been really fun. It was great to do this.